Hello, my name is Ksenia Cohen-Katzenelson, and I'm the group leader of San Diego R&D team with Eurofins Discovery. Today, me and my colleague, Alison O'Mahony, who is the VP of Translational Biology and Phenotypic Services, will tell you how we approach targeted protein degradation at Eurofins Discovery, from both chemistry to consequence. I will present about the E3 scan target-based platform, and Alison will present about the BioMAP phenotypic profiling platform. The agenda for today will start with a brief introduction about targeted protein degradation and overview of Eurofins Discovery services and products in this space. Then I will dive into the E3 scan technology and assay validation. After that, Alison will speak about the BioMAP phenotypic profiling of compounds in TPD. Targeted protein degradation, or TPD, is the use of small molecules to hijack the cellular degradation machinery by recruiting E3 ubiquitin ligases to proteins of interest to induce their ubiquitin-dependent degradation. TPD is of interest in drug development as it can address previously considered undruggable targets such as scaffold proteins or transcription factors, which constitute about 80% of the human proteome, and those cannot be inhibited with traditional small molecule inhibitors. It's been 20 years since the introduction of the first PROTAC degrader molecule. However, degrader discovery and optimization remains an empirical, slow process. The majority of the degraders that are in clinical trials use mainly the cereblon or VHL E3 ligases to ubiquitinate a protein of interest. Recent studies suggest that varying the recruited E3 ligase can influence the degradation of the target space. About 300 out of these 600 human E3 ligases are predicted to be involved in the ubiquitin proteasome system. So by adding these to the TPD toolbox, the degradable target space can be significantly expanded. To accelerate the TPD field, we are expanding the TPD toolbox by developing additional E3 ligase target assays. Here at Eurofins Discovery, we offer a variety of services and products for TPD drug discovery space. To detect binding to E3 ligase, we have the biochemical E3 scan assays that I will tell you more about shortly. To detect binding to a target protein, we offer the kinome scan, bromoscan, and BCL2 scan assays. We also offer cell-based assays to assess target degradation or engagement, and BioMAP platform for phenotypic profiling that Alison will tell you more about it. We also offer chemistry beyond the rule of 5. If you're interested in any of these additional services, please reach out to our booth or email us directly. And now I'll tell you more about the E3 scan platform. We have previously applied the kinome scan technology to develop substrate recruitment site-directed competition binding assays for diverse E3 ligases. Using this kinome scan technology, we have developed in the past almost 500 assays for kinases, 40 assays for bromodomain proteins, and 5 assays for the BCL2 family. And using the same technology, we now develop 9 assays for different E3 ligase targets and assays for the individual beer domains for substrate selectivity screening. The E3 scan assay principle is described here. It is very similar to the kinome scan technology and has 3 key components in the assay. As you can see in panel A, the first component is an E3 ligase that has a DNA tag on it and is expressed using our proprietary mammalian or phage display expression systems. This is not a purified protein, but a protein extract, which makes it very easy and fast to make, and scalable. The second component is a ligand for the E3 ligase that is immobilized on a solid support bead. And the third component is a test compound or a control solvent. The 3 components are incubated together, the beads are washed and eluted, and the elution is then used for an ultrasensitive qPCR readout. In panel A, what happens in the absence of a test compound? The E3 ligase binds to its ligand on the beads, the wells are then washed and eluted, and therefore the signal of the E3 ligase is then detected using the qPCR readout. So in this case, the signal will be high. In panel B, in the presence of a test compound that can bind to the E3 ligase and compete it off the beads, the E3 ligase will be washed off and will not be detected in the elution using the qPCR. So therefore, the signal will be low. In panel C, if the test compound does not compete the E3 ligase of the ligand beads, the E3 ligase will again be eluded in the assay and the qPCR signal will be high, just like in panel A. So we are looking for a loss of signal in the assay. We have previously launched E3 scan assays using several well-studied E3 ligases that are often used in TPD. Here are shown representative control ligand curves of these targets using our E3 scan assays. You can see here is example of a simple class of E3 ligases such as MDM2 and its partner MDMX, which has no known E3 ligase activity. Here are shown complex E3 ligases, VHL and cereblon, that require partner proteins for their activity. And here are the beer domain E3 ligases, CIAP1, 2, and XIAP. All of these assays are done using human full-length protein. As you can see, all of these assays are robust, high throughput, and show correct potency in rank order for the control compounds we tested. The assays can also detect picomolar KDs for small molecules and peptides. In addition, I wanted to add that for the full-length beer domain E3 ligases, we now have available individual beer domain assays. This allows for selectivity screening of ligands against the different domains. If you are interested in the beer domain data, it is presented at our poster in this meeting. Using these 7 established targets as a proof of concept for E3 scan, we looked into developing assays against additional E3 ligases that have no known published small molecule ligand. In this simple E3 ligase tree, we chose a subfamily of HEC-NED4 E3 ligases. This subfamily includes 9 members, all involved in the proteasome degradation system, therefore can be potential target in TPD. In addition, the entire subfamily shares similar domain structure whereby these E3 ligases recognize their substrates through their conserved domains. And even more attracting for us was the fact that the WW domain likes to bind a Ppxy motif in its substrate. The Ppxy motif creates a potential opportunity to develop an E3 scan assay without using a small molecule bait, but rather a peptide bait that has the Ppxy motif in it, and will be able to capture the E3 ligase on the bead in our assay. Here is shown E3 scan assay for the human WW domains 1 and 2 of each. Using a bait ligand peptide that contains a Ppxy motif in it, we were able to establish an assay window of 300-fold and measure binding affinities to several Ppxy motif peptides that we collected from the literature. As expected, we observed potent binding of each to its substrate motif peptide. We have also observed potent binding to the substrate motif of NED4. Similar to each, here is shown an E3 scan assay for the human W domains 2 and 3 of NED4. In this assay, we used similar strategy with a bait ligand peptide to the one we used in each assay with a Ppxy motif in it to establish an assay window of 200-fold and measure binding affinities to several Ppxy motif peptides. As reported in the literature, we observed potent binding of the NED4 substrate motif peptide, and also, in this case, binding to the each substrate motif, just like we've seen the opposite for each. For the other substrate motif peptides, we observed weak or no binding for both each and NED4, as I will show you shortly. This suggests that each and NED4 have substrate selectivity among the Ppxy motifs. This table summarizes the results from screening the Ppxy motif peptides against each and NED4 E3 scan assays that we developed. The Ppxy motif peptides we used are derived from known substrates of the NED4 subfamily. For example, if you look here at the each peptide, it has two Ppxy motifs from its substrate TXNIP. This peptide show potent binding to each, just as we expected, and in addition, we saw also potent binding to NED4. The NED4 1 and 2 peptides are derived from the NED4 substrate ARRDC3. And the only difference between them is that one has two Ppxy motifs and the other is just one. The peptide with the two motifs showed stronger binding to NED4 as expected, and in addition, we also observed binding of these peptides to each. So this suggests that each and NED4 have common preference for their Ppxy motif substrates. The WBP peptides that we tested are derived from a YAP ligand, which also has been shown to target its substrates through its WW domains. In the literature, these peptides were shown to bind another NED4 family member, WWP1 and 2. However, for each and NED4, we observed very weak or no binding. The SMAD7 peptide was derived from the SMAD7 adapter of SMARF2, another member of the NED4 family. This peptide also shown weak or no binding to the each and NED4. The ENaC beta wild-type peptide is derived from the epithelial sodium channel substrate of NED4. This peptide, although shown to bind the rat NED4 in the literature and several other NED4 family members, we did not see binding in our human assays for these peptides. So overall, there seems to be selectivity among the NED4 family members to different Ppxy motif substrates. We are currently in the process of developing assays for the rest of the NED4 family members, and it will be very interesting to see their binding to these peptides as well. So in summary, we have developed and validated E3 scan assays against additional E3 ligase targets that have no known published small molecule ligand. All of these assays are robust, high throughput, and give high-quality KD curves. The assays can support an SAR campaign with rapid turnaround times for library screens of 20 days and weekly SAR with 5 days turnaround time. And it is the largest E3 ligase panel available on a single technology platform. We have more assays in development, and we are not limited by having a ligand small molecule, as I have shown you here. And now my colleague Allison will tell you how you can supplement your degrader drug discovery and development using the Phenotypic BioMAP platform.
Thanks, Ksenia. We're now going to change gears to introduce the BioMAP human phenotypic platform and discuss how it was used to evaluate targeted protein degraders based on their impact on human tissue and disease biology, and how such activities and insights can help guide drug discovery and development efforts in this innovative space. The BioMAP phenotypic platform was developed in response to a need for better, more translational in vitro models of human diseases to support drug discovery. The three main pillars of the platform include the biological assays, the analytical tools, and an extensive reference database. BioMAP systems are standardized in vitro human disease models, comprised of primary cell types pooled from multiple healthy human donors, used at low passage to preserve high physiological relevance, and stimulated with disease-relevant factors such as cytokines or receptor ligands. These assay systems are used to measure levels of clinically relevant protein biomarkers following treatment with compounds or controls. The resultant data is visualized in mind using our proprietary and predictive analytical tools to gain insights on mechanism of action, identify efficacy and safety-related biomarkers, and enable comparative benchmarking analysis. The BioMAP reference benchmark database contains activity profiles for more than 4,500 non-proprietary agents, including clinical drugs, failed candidates, and other benchmarks representing both small and large molecules. This database serves as a repository for thousands of mechanistic signatures that are leveraged to analyze and interpret the activities of new compounds. BioMAP testing is offered in two formats: profiling and early screening. BioMAP profiling is designed to provide a deep and comprehensive characterization of more advanced or later stage compounds, with analysis and interpretation from our reference database to provide key insights on biological outcomes. Early screening services involve more streamlined processes and deliverables designed to test larger numbers of earlier stage compounds in order to rapidly de-risk, debulk, and differentiate chemistry. In this case study, we profiled a number of compounds targeting the BET family of bromodomain proteins. Using the BioMAP Diversity Plus panel, we assessed their impact on translational protein biomarkers in several in vitro disease models. Before we discuss the data, I'd like to orientate you to a BioMAP Diversity Plus profile. Along the top are the 12 individual BioMAP systems that make up this panel. Each system is comprised of different human primary cell types, cultured alone or co-cultured to recapitulate aspects of tissue and disease biology. Systems are treated with compounds or controls and are then stimulated for 24 hours or longer. Along the x-axis are the 148 biomarker readouts, including both cell-associated and soluble protein biomarkers, as well as proliferation and cytotoxicity endpoints. The y-axis represents the relative expression levels of each biomarker in the presence of the test agent, normalized to the parallel tested vehicle controls. The averaged vehicle control for that experiment is represented along the 0 line, and the gray envelope surrounding this is the historical vehicle controls, which serve as a 95% confidence interval to identify biomarkers of the test agent at 4 concentrations. Here we show the annotated and analyzed profile for the bromodomain protein degrader MZ1. This agent was profiled at several concentrations, but had detectable cytotoxicity in multiple systems at or above 120 nanomolar. At these lower concentrations, we note that MZ1 still has cytotoxicity in one system modeling macrophage activation that's relevant for innate immunity. The gray arrows indicate that MZ1 also inhibits the proliferation of T cells and B cells, but does not impact the proliferation of other adherent cell types such as endothelial cells, coronary artery smooth muscle cells, or fibroblasts. We can see by the annotated activities that MZ1 modulates a broad scope of inflammation, immune, matrix remodeling, and hemostasis-related biomarkers. A comparison of the profiles for MZ1 at 41 nanomolar and an equimolar concentration of a selected benchmark cis-MZ1 revealed a total of 73 differentiating activities, consistent with the effects of an active targeted protein degrader MZ1 that are not seen with the negative control cis-MZ1. However, 4 common activities were identified as annotated on this plot. These indicate the shared effects of the common scaffolder structure of these two molecules. In an unsupervised search for mathematically similar profiles for MZ1 from the thousands of profiles in the BioMAP reference benchmark database, the most similar match was its target-related bromodomain inhibitor, JQ1. We identified 17 common activities between these profiles, which represents a potential bromodomain negation signature. The Pearson correlation coefficient was just below our determined threshold for mechanistic similarity, indicating that these molecules can indeed be differentiated. 15 differentiating activities, as annotated on this plot, separate the distinct mechanism of actions for these two compounds. Additional similarity hits for MZ1 from the database included other bromodomain degraders and inhibitors listed here. This prompted us to expand the evaluation and comparative analysis for other molecules within the TPD class using the same Diversity Plus panel. Here we show an overlay of the BioMAP Diversity Plus profiles for 3 such molecules at their indicated concentrations. Comparative analysis reveals that they share 10 common activities. These activities include inhibition of immune and inflammation, as well as matrix remodeling response biomarkers. This shared signature suggests a development path for autoimmune or inflammatory indications. Comparison of the profiles for two E3 ligase modulating compounds, pomalidomide and lenalidomide, reveal 4 annotated activities, annotated in green. These include effects only in immune cell-based models. The common activities indicate that pomalidomide and lenalidomide both inhibit B-cell activation, consistent with clinical use. 8 differentiating biomarkers were also identified, annotated here in black, and reveal that pomalidomide is more active than lenalidomide at the same concentration, which is also consistent with clinical reports. Network clustering analysis was used to determine the similarity of all compound profiles at multiple concentrations. Here in panel A, each dot represents a specific compound at a test concentration. Connected dots indicate that their BioMAP profiles are mechanistically similar. We show that molecules targeting the bromodomain proteins, either by small molecule inhibition or by targeted degradation molecules, cluster with themselves, but they do not cluster with their E3 ligase compounds. In panel B, the bromodomain cluster can be separated at a higher, more stringent Pearson correlation cutoff, and the resulting clusters bin into either bromodomain inhibitors or degraders. This type of analysis can be very useful in differentiating multiple exemplars from an SAR program, for example. The analysis that I've shown thus far represents BioMAP profiling studies. Early screening services involve a more streamlined process and deliverables designed to test larger numbers of earlier stage compounds. The goal of diversity early screening is to rapidly de-risk, debulk, and differentiate chemistry based on the biological outcomes achieved in this platform. Diversity early screening uses the same 12 systems described earlier for the Diversity Plus profiling study, but are used here to screen a minimum of 72 samples, where a sample is one compound at a concentration. Streamlined analysis and deliverables include a heat map of biomarkers, a summary dashboard of hits and toxicity signatures, and a similarity clustering to compare samples. Key points on profiling targeted protein degraders in the BioMAP platform include the ability to compare molecules within or across classes and identify and interpret the biological outcomes. We showed that while the protein degrader MZ1 was significantly more active than its negative control cis-MZ1, the negative control was not without biological activity. This means that BioMAP profiling can identify primary and secondary pharmacology to prioritize scaffolds, rank candidates, and provide additional unbiased translational information to advance hits. Bromodomain degraders were cytotoxic to human primary cell types at concentrations close to reported EC50 in assays that do not address cytotoxicity. These insights are crucial to the selection and development of safe and efficacious therapies in this degrader space. Finally, highly reproducible BioMAP data allows for similarity analysis to discriminate inhibitors from degraders or to differentiate molecular structures or other characteristics. To summarize the take-home message presented by Ksenia and myself, Eurofins Discovery has a comprehensive and growing portfolio of solutions and services for targeted protein degraders. Offerings include a large suite of target-based, cellular, and phenotypic platforms that can prioritize candidates with high therapeutic potential. Our E3 scan ligand binding platform can identify and characterize new, potent, and selective ligands that bind and reprogram E3 ligase substrate specificity. Our BioMAP phenotypic profiling and screening platform facilitates unbiased evaluation of targeted protein degraders or other compounds for impacts on translational biomarkers relevant for efficacy, safety, candidate prioritization, and guidance on indication selection. We thank you for your attention and we will be happy to take questions.